All right, saints. We are back. We are back again. All praise is due to the Most High God. This is the day that the Lord has made. I will rejoice and be glad in it by the grace and mercy and just the great works of God. I am back here ready to do part three of the GOAT series, the greatest hack of all times. So let's go ahead and jump right in. When we left off, on the second video, we were talking about the Nimrod protein. And so, in regards to this Nimrod protein, as we were seeing references to this process, we saw this word bacteriophage come up. We need to figure out what bacteriophage is. So, let's go ahead and see what bacteriophage is. Um, Now it says bacteriophage, also known informally as phage, is a virus that infects and replicates within bacteria and archaea. The term was derived from bacteria in the Greek phagian, meaning to devour. All right. So we're getting, again, if you recall, we were getting iterations of eater, ingestion, and now we're getting a root meaning from the Greek word phagian, which means to devour. Um, Bacteriophages are composed of proteins that encapsulate a DNA or RNA genome and may have structures that are either simple or elaborate. Their genomes may encode as few as 4 genes and as many as hundreds of genes. Phages replicate within the bacterium following the injection of their genome into its cytoplasm. This is artistic depiction of what a bacteriophage is, a virus rather, um, and the process that it does for the injection of these viruses into a cell. All right. Now, uh, take a look at this thing here. Again, these spider-looking things. I'm at work right now, so it's a pretty controlled environment here. They have the Orkin guy come out and spray this thing out the wazoo. So I'm good to go here. But even looking at this thing, it's very unsettling. All right. Now, let's see here. So one second here. Let me see if I can reference something in here. All right. So what I'm supposed to reference in here is this, and I will highlight it here for you. Okay. So meanwhile, bacteriophage researchers have been developing engineered viruses to overcome antibiotic resistance and engineering the phage genes responsible for coding enzymes that degrade the biofilm matrix, phage structural proteins, and the enzymes responsible for lysis, which is defined as the breaking down of the membrane of a cell, often by viral, enzymic, or osmotic mechanisms that compromise its integrity. Uh, and the enzymes responsible for this lysis of the bacterial cell wall. There have been results showing that T4 phages that are small in size and short-tailed can be helpful in detecting E. coli in the human body. All right. Um, So this process called bacteriophage that is achieved by um, how a virus injects itself into a cell. I have heard it a more crude explanation of the process being related to the raping of a cell. um, the raping of a cell, pretty much. Now that's some pretty invasive terminology that I just used, and I don't like using it given the connotation behind it. Um, but just to give some context on what this process is, um, if you can recall, if we've even covered this video yet, which I think should be coming later on, the Jennifer Doudna, the CRISPR co-creator, there is a segment in that video that I do the commentary on to where you can see this process where it injects from here, and it almost looks like a little squiggly semen looking thing that injects in there. It's very reminiscent of what is done in being fruitful and multiplying, so to speak. Let's just keep it in the verbiage biblical, all right? Um, so what they're referencing here, uh, again, I'm sorry, I've got this mouse here. I'm actually at work, so I'm pretty much working in reverse of my screen, so I've got to get used to it. What they're talking about, this where was that? There have been results showing that T4 phages that are small in size and short-tailed can be helpful in detecting E. coli in the human body. This T4, we need to go check out what T4 is, which brings us to this, the Escherichia virus T4. Now again, we get this freaking thing right here. This is, oh my goodness, yeah. Let's get off of that. Now, uh, this right here, it says we'll just say T4 virus is we see it here, Escherichia, we'll just say E virus T4 is a species of bacteriophages that infect E. coli bacteria. It is a double-stranded DNA virus in the subfamily. I'm not going to try that. We'll just call that Tevin from the family Myoviridae. Myoviridae. Wife, I know you're laughing at me right now. T4 is capable of undergoing only a lytic life cycle and not the lysogenic life cycle. The species was formerly named T. even bacteriophage, a name which also encompasses, among other strains or isolates, Enterobacteriophage T2, Enterobacteriophage T4, and Enterobacteriophage T6. Um, this right here, I need to reference this. T4 is capable of undergoing only this type of life cycle. Is one of the two cycles of viral reproduction, the other being the lysogenic. So this one results in the destruction of the infected cell and its membrane. Bacteriophages that only use the lytic cycle are called virulent phages, um, and not this one. So this T4 virus is one injected into the bacteria or the cell, results in the destruction of the infected cell and its membrane. Now, um, one second here. Now, the thing is, this T4 virus, okay? This T4 virus. Let me take a sip real quick. Before we go for it, here's another. This looks like more of an in vitro rendition of it. So that, yeah, that looks like it's under a microscope, like it's been multiplied under a few times under a microscope to get this picture taken. And again, we get this almost hexagonic shape, like an oval hexagon shape, kind of just stretched out, and the body, the tentacles, and we'll say the delivery right there, right? Let me see if there are any other. Yeah. Let's go ahead and take a look at this here real quick. So this is the head. This is the collar. Maybe that's the collar, and this is the tail. Long tail fibers, and base plate. Oh, look at that. Now that's the DNA. That's the protein. And here you see the injection, and pow, it comes right out of there. All right, yo, that's a that's a la- that's a penis in a laboratory. Let's just go ahead and keep it 100. All right. This is yeah, yeah. It's reminiscent of, especially of what it's what it does. That's the DNA protein strand in there that gets injected out that way. All right. 1 second here, and I just want to make sure there aren't any other. Yeah, we're good to go. So this T4 virus. All right. You see that? Okay. Does that remind you of anything? Does that remind you of anything? Tyrant virus from the Resident Evil series. Tyrant virus. Tyrant virus, otherwise known as the T virus, is the general name given to a series of mutant progenitor virus strains. All right. Um, this is from the Resident Evil series. Okay. Um, interesting. Now, interesting being that Nimrod is generally depicted as the first tyrant and empire builder in the Bible. All right. This tyrant virus. All right. Keep this in mind. See Nimrod popping up a few times in this. So just keep that in mind here. Um, we will go here. This is let's see, October 12th, I think it was, circa 2020, and this is in regards to the bacteriophage T4 virus again. Um, and we're just getting just getting a little bit more information on it. That infecting bacteriophage T4 uses a contractile contractile tail structure. I just see contract, all right, but contractile tail structure to breach the envelope of the E. coli host cell. Now, this um, breach the envelope, that's not doing this process any justice. It almost desensitizes you with the verbiage. But um, if you watch the Jennifer Doudna CRISPR co-creator video, you'll see what what that process is. All right, when I said it's raping of a cell, that's exactly what it looks like. It's very invasive. All right, um, but getting back to what it says here, during contraction, again another word association to birth. Um, in regards to what this if you inseminate something in regards to injecting DNA, that's pretty much you're injecting seed, so to speak. And so you have this word association uh, during contraction. The tail tube headed with the central spike complex is thought to mechanically puncture the outer membrane. Um, and so this is just again, that's the DNA. Interesting that they have it in the form of a spiral, and this is where the insemination happens. All right. Now, what you don't see below here is the actual membrane or the cell or the bacteria that it's injecting. But when I say um, it's unsettling to see that process. Um, Now, we've seen E. coli come up in regards to thymine and mutagenic changes as a result of of inaccess or decreased amount of thymine in the ACTG sequence. Let's take a look at something here. Now, this is July 16th, circa 2017, a living hard drive. This GIF or GIF was stored in the DNA of bacteria. Now, let's see, DNA is a hugely promising medium, that word medium, for storing data. Consider that a cell nucleus can hold the instructions for an organism as complex as a human. So far efforts to store non-genetic data and DNA have been carried out in test tubes. But now scientists have encoded a GIF or GIF into the genome of living bacteria. The scientists from Harvard University used the CRISPR genome editing tool to store a picture of a hand and an animation of a running horse adapted from Edward Moy Bridges' 1878 photographic study, Human and Animal Locomotion, all stored in the genome of E. coli bacteria. So here's the very interesting here. I want to show you this. So this moving image right here that you're seeing, this was one of the images or moving locomotive pictographs that they were able to store in the DNA of bacteria, specifically E. coli. Um, they were able to understand when they say they were able to upload it, and then they were able to extract this from the DNA. Um, that's that's absolutely crazy that they were able to do that. Um, I'll go on further here. Efforts to store unconventional data in DNA have been going on for years, thanks to DNA's incredible compactness and long shelf life. Now, if you recall, I think they said in just a strand of human DNA, just a bit, a prick of DNA from a human is the equivalent storage capacity of 7 million iPads, I think it was. It was either 7 million or 7 billion. I apologize. That million and billion, there's a huge gap in between that, but still, nonetheless, the storage capacity of wow, 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 wow. Um, Typically, though, this has been done by translating the bits that encode books, images, or audio recordings into DNA sequences and then synthesizing them artificially by using CRISPR. Instead, the Harvard team, led by renowned geneticist George Church, was able to hijack the genomes of E. coli bacteria to store the information. This George Church, he actually comes up in the when I was telling you about the storage capacity of DNA being compared to the storage of 7 billion iPads or 7 million iPads. This George Church is actually in a commentary that I'm doing for this series for Dr. Charles Morgan, where George Church's revelation was brought up. Well, not revelation, but research was brought up. And so that's just giving you some background into this, but I want to show you. Remember, they said, let's see, to store a picture of a hand and an animation of a running horse. Now, up here we have 4 horses, and they all look different. All right, this one has blotches, this one is darker, and this one has blotches and different. So they're 4 horses. Saints, uh, what is this reminding us of? All right, what is this reminding us of? Now, understand these references to CRISPR. Okay, um, it says, let me go down further in here. The CRISPR system is actually a natural defense mechanism that bacteria use to develop immunity to invading viruses by recording snippets of the attacker's DNA in the bacteria's genome. These snippets are then used to guide the enzyme Cas9 to find and destroy invasive DNA next time the virus attacks. Um, so the thing is, or they're saying this CRISPR, I mean, it's a natural function of E. coli. Um, but the CRISPR itself, they just named the function, okay? In this process, they named the function that this uh, uh, E. coli has within it. And they just named it CRISPR, and it's an acronym for something. I know who is it? Jennifer Doudna um, says what the acronym is in the video commentary that I've got for this series. Be sure to reference that. Taking a drink. Unfortunately, I can't reference it offhand. That's just not in my repertoire for that. Um, but one second here. Now, to take it a step further here with this, keep this in mind, okay? And then the hand, all right? Store a picture of a hand. This is BBC News in regards to the same research. July 13th, circa 2017. Again, we get a very pixelated image of this. An image in short film has been encoded in DNA using the units of inheritance as a medium for storing information. All right. Uh, and remember the hand that they talked about. This is the pixelated image of a hand. Notice it's the right hand. Notice it's the right hand, you guys. Um, what does that remind us of? All right. What does that remind us of? The four horsemen. Well, excuse me. Well, I just said it. The four horsemen of the apocalypse, and also this right hand. What does it say in the Bible? All right. We know what it is. So we're going to let's see. 1 second. Now we have to understand the relation. How does this relate to us in regards to the E. and this CRISPR software and the editing images into E. All right. We go to this. Let's see, October 16th, circa 2020. Photon directed multiplexed enzymatic DNA synthesis for molecular digital data storage. All right. This is letting us know. This is just more emphasis on the properties of being able to store information in DNA. All right. And here we're talking about photon uh, directed multiplexed enzymatic. So it says here, uh, due to its stability, information density, and ease of readout with advanced sequencing techniques. So this is all in regards to what CRISPR is able to do. Um, However, progress in writing DNA is stifled by the continued reliance on chemical synthesis methods. All right. That's just giving us a little bit more perspective here, but pretty much just reiterating what we saw in the last two links. Now, here's the thing. This is where the link comes in as far as that, what it has to do with us. I don't want you. No, come on. I don't want you. Oh, no. It says I've reached my limit for free articles. Oh boy, oh boy, oh boy, oh boy. Let me see something here. Let me see. What are we looking for? Okay, well, you know what, Saints, you're gonna be able to look this up for yourself because I've looked at this article too many times it's saying, but I can read to you basically what it says. Now, this article is called "Link Between Human Genes and Bacteria is Hotly Debated by Rival Scientific Camps," the New York Times reports. Now, what's outlined in this article that we can't see, and then other research papers that you can reference. I can't pull up the white papers because it's going to mess with all of my links here. But basically, I'm gonna give you a summation here in my notes. What needs to be understood here is that they found that the genomic and neurological database of bacteria, the memory where it talks about it, able to um, keep information as far as attacking and defending itself against attacks from um, um, foreign things. Um, what needs to be understood here is that they found that the genomic and neurological database of bacteria, specifically E. coli, mirrors the neurological synapses of a human brain's functionality, especially in the memory spectrum known as the hippocampus of the brain. Anybody in medical, you've heard of HIPAA, all right? So the hippocampus, they pretty much named it that because it's protected and valuable information in regards to how and where and why the brain stores it. Um, especially in the memory spectrum known as the hippocampus of the brain, basically saying that the human's body and brain's way of storing information and memory for neurological functions and survival functions in our immune system is very similar to how bacteria stores information. Again, another alternate process utilized to duplicate a process already in place, which is another signature, yet again, of the enemy. We will revisit this information in a major way later on in the series. Now, we had a reference to something called CRISPR in regards to the image being made, being able to be stored in DNA. What is CRISPR? Let me jump back over. Jumping in between screens sometimes. The mouse, you gotta like beat it over. I'm sure you guys know what I'm talking about. Come on, hold on, get over there. Come on now. Gotta be real. There we go. You almost gotta like shoot it into the next screen. What is CRISPR? CRISPR, a segment of DNA containing short repetitions of base sequences involved in the defense mechanisms of I'm not doing it right now. Organisms to viruses. Now, this is the process that they're talking about, the natural process that exists in bacteria. Um, they named this process CRISPR. Now, uh, CRISPR is a genetic engineering tool that uses a CRISPR sequence of DNA and its associated protein to edit the base pair of a gene. This is basically gene editing software, a gene editing program. All right. Um, Now, meet Jennifer Doudna, who I just referenced above. So yeah, I'm right on track, Lord. Thank you. I thought it was further down, but now we get to meet this Jennifer Doudna. She is the co-creator of CRISPR. Now, the Lord reminded me that with all of the technical and medical jargon these folks use, can obscure the enemy's true intentions, uh, which can be considered high forms of doublespeak and code in regards to the medical jargon that they use. This in turn led me to do a live, summative, and simple translation of a few videos to decode what these people are saying, with this video of Jennifer Doudna being the first in this series. A lot of censorship is going on, so I had to upload this video as unlisted, but it will be provided in the links with this series for you to watch at your own leisure, and as to not lengthen the content of the main video that you're currently viewing. Um, so I will be including that video down in the description of uh, um, of this main video that you can watch, and you can get a very good gauge and grasp on what this technology is, the lady behind it. But I'm going to dive into this lady here real quick for us, all right? But just be aware that what CRISPR is. I've got a video commentating on it, and I will be providing that in the description of the video. Now, we need to go a few places here real quick, guys. CRISPR, uh, Cas9 helps uncover genetics of exotic organisms. All right. Um, and this is just giving us some good background. The simplicity of CRISPR Cas9 gene editing will soon make studying the genes of any organism, from the simplest slime mold to the octopus, as easy as it, excuse me, as easy as it now is to study the genes controlling development in standard lab animals such as nematodes, fruit flies, frogs, and mice. All right, remember the fruit flies. So right, remember the fruit flies. So that's just giving us a little bit of background. This is December 10th, circa 2015. Um, we'll go a step further here. Just a little bit more supporting information on it. CRISPR mediated modular RNA guided regulation of transcription in I can't eukaryotes. I can't mention freaking words. God, thank you, Lord, for helping me here. Now understand some of these words, man. This is July 18th, circa 2013. The genetic interrogation and reprogramming of cells requires methods for robust and precise targeting of genes for expression or repression. These terminology that when I said this technology is invasive, interrogation, like they're holding cells at ransom, like, show me what you do. You're gonna do this now. Where's the bomb? In the Jack Bauer voice, where's the bomb? But this interrogating or interrogation and reprogramming requires methods for robust and precise targeting. I mean, the implications in the verbiage used should alarm you that it's unnatural. It's not supposed to be. This isn't this. It's not right. All right. Um. So 1 second here. Did that. So where did we get this from? I need to check. I need to check here real quick. Okay, this is where we got it from. All right. So here I had to go back. I had to go back real quick. A new study from UC Berkeley illustrates the ease with which CRISPR-Cas9 can knock out genes in exotic animals. In this case, an amphipod or sandhopper. Now, if you watched, I think it was in the first trailer. I think it was in the first trailer for the Goat series. You saw, you saw this. You saw this. All right, amphipod. All right, and with the what is this? You can see it here. This is the body, and then the different colors: the yellow, blue, and the purple, which represent them scanning electron microscope images of a normal amphipod about 1 millimeter long, with color-coded hind jumping legs, and an amphipod that has been genetically modified with CRISPR to grow forward walking legs instead. Um, and so this is how they were, and they highlighted them for reference. And when they reprogrammed it, I mean, you see they were going back this way, and here now they've got him walking forward. These legs pointed forward, and that's just with the gene editing software. Lord wanted me to. What is an amphipod? Right? Amphipod is an order of blah blah with no carapace and generally with laterally compressed bodies. Amphipods range in size from blah 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 and are mostly bees or scavengers. There are more than 9,900 amphipod species so far described. They are mostly marine animals. Marine animals, again, another ode to aquatic life, but are found in almost all aquatic environments. Again we get more references to marine kingdom organisms. Now check this out. We've got to jump back here. This Jennifer Doudna, okay? This Jennifer Doudna. Forgive me. I actually probably need to get some caffeine because it is late, as you can probably see right here, and I feel myself drowsing out. But thank you, Lord. I can do all things through you, through Christ Jesus, which gives me strength. Amen. Hmm. So, haunting, Doubtnut, nightmare about Hitler wanting CRISPR. Say what? Say what? Now, this Jennifer Doudna, again, is the co-creator of CRISPR. Show you something. A recent piece on CRISPR genetic modification in the New Yorker called The Gene Hackers, or Human 2.0. Come on, y'all. Come on. Whatever happened to Human 1? The one that God created. We got 2.0. That's an ode to transhumanism, y'all. You know what that is. 2.0. Part 2 human, you know. But so let's see. The article, 1 second. The article provides an in-depth look at CRISPR and its potential use for the human editing. So she was interviewed by the New Yorker, and during the interview, she had something to say. Um, she says, the thing that struck me the most was the recounting of a dream, really a nightmare, that left the CRISPR pioneer Jennifer Doudna waking up in a cold sweat. In the nightmare, Adolf Hitler wants to learn more about CRISPR and presumably to use it for eugenics. You know what eugenics is, y'all? All right? The eugenics is basically, CRISPR is basically the brainchild of gene editing software that, of eugenics. of eugenics that came, that Hitler pretty much, I won't say he started, but he definitely uh, catapulted, in his that evil reign that he had. Um, this dream can be seen as a reflection of the disturbing possible future reality that some may take CRISPR technology in unethical directions, and there could be not a whole lot that any of us, including a leading scientist like Dr. Doudna, could do to stop them. So we're gonna I think, yeah, we get to go to it. Um, and what I need to do. So this is the New Yorker. This was November 8th, 2015, as you see. And so we'll set it up. It says Doudna, that's Jennifer Doudna, is a highly regarded biochemist, but she told me that not long ago she considered attending medical school. Uh, uh, she considered attending medical school or perhaps going into business. I'm going to highlight everything here. She said that she wanted to have an effect on the world and had begun to fear that the impact of her laboratory research might be limited. The promise of her work on CRISPR, however, has persuaded her to remain in the lab. She told me that she was constantly amazed by its potential, but when I asked if she had ever wondered whether the powerful new tool might do more harm than good, she looked uncomfortable. She says, I lie in bed almost every night and ask myself that question, she said. When I'm 90, will I look back and be glad about what we have accomplished with this technology, or will I wish I'd never discovered how it works? Her eyes narrowed. Ah. Her eyes narrowed, and she lowered her voice almost to a whisper. I have never said this in public, but it will show you where my psyche is, she said. I had a dream recently, and in my dream, she mentioned the name of a leading scientific researcher. Um, this scientific researcher had come to see me and said, I have somebody very powerful with me who I want you to meet, and I want you to explain to him how this technology functions. 1 second. So I said, sure, who is it? It was Adolf Hitler. I was really horrified, but I went into a room and there was Hitler. He had a pig face. He had a pig face, and I could only see him from behind, and he was taking notes, and he said, I want to understand the uses and implications of this amazing technology. I woke up in a cold sweat, and that dream has haunted me from that day, because suppose somebody like Hitler had access to this. We can only imagine the kind of horrible uses he could put it to. Saints, saints. Hitler, in her dream, and he has a pig face. Let's go a step further. All right, play this.
Are you an IT pro? So you must be using all of these tools. Remote monitoring.
Well, CRISPR is an acronym that actually represents a sequence of DNA letters in the genomes of cells. It's found in bacteria, and it was interesting to scientists originally because it's a bacterial immune system, a way that bacteria can fight viral infection. But the CRISPR acronym has now become widespread in the media as an indication of a new technology for gene editing. And the story of how an adaptive immune system in bacteria was harnessed as a technology for gene editing is really part of what a crack in creation is about. So the CRISPR gene editing technology is a tool that scientists can use to change the letters of DNA in cells in precise ways. So I like to use the analogy of a word processor on our computer. So we have a document. You can think about the DNA in a cell like the text of a document that has the instructions to tell the cell how to grow and divide and become a brain cell or a liver cell or develop into an entire organism. And just like in a document, the CRISPR technology gives scientists a way to go in and edit the letters of DNA, just like we might cut and paste text in our document, or replace whole sentences, even whole paragraphs or chapters. We can now do that using the CRISPR technology in the DNA of cells. So when we think about a technology that allows precise changes to DNA to be made, for scientists, this is sort of really a gift that allows research to proceed very quickly in terms of understanding the genetics of cells and organisms, but also provides a very practical way to solve problems. And there's many that we could discuss, but I'll mention a couple that I think are particularly exciting. So in clinical medicine, the opportunity to make changes to blood cells that would cure diseases like sickle cell anemia, a disease where we've understood the genetic cause for a long time, but until now there hasn't been a way to actually think about treating patients. And now with this technology, it's possible in principle to remove stem cells that give rise to blood cells in a person's body, make edits to those cells that would correct the mutation causing sickle cell disease, and then replace those cells to essentially give a patient a new set of cells that don't have the defect. So I think that's very exciting, and there are multiple research groups right now working on doing just that. So I think that's a future, probably sometime in the next 2 to 5 years, we will see clinical trials in that area, and we hope real progress towards curing that disease. But there's another example that I think is also potentially very impactful clinically, but has a very different kind of strategy, is the idea of making edits to pigs to create animals that are going to be better organ donors for humans. And so pigs are already of interest for organ donation, but imagine that we could make edits to the DNA of pigs to make their organs more human-like, and also to remove any viruses from pig cells that might otherwise infect a patient. And those are both things that are actively underway using the CRISPR technology. And then a third area that I think is interesting to think about from the perspective of global impact in disease is thinking about using gene editing not to change the DNA in people per se, but actually to affect the kinds of insects that transmit disease to people. And the idea here is that one could use a gene editor to create mosquitoes that would be unable to transmit viruses like dengue virus or Zika virus by using a technique called gene drive that allows traits to be spread very quickly through a population using an efficient way of gene editing, such as the CRISPR tool. And I think that's an opportunity that could have very big impact in terms of global health, but also requires obviously some very thorough vetting and discussion about potential environmental impact. I think one of the aspects of this technology that's been very interesting to me personally is my own kind of personal growth through the last few years. I think that, when I started this research project, which actually began now almost 10 years ago in the lab, we were certainly not thinking about technology that would allow alteration of human evolution or anything of that nature. And over the last few years, as this technology has begun to be deployed globally for different applications, I've found that I've gone from thinking about it initially just with sort of almost wide-eyed excitement, thinking about all the opportunities that this offers, to realizing that there was real risk, and that we really needed, we meaning the scientific community and really frankly the human community, needed to be aware of this and discussing it. And one of the things that sort of brought that to the forefront of my mind was a dream that I had fairly early on, in which I was, I walked into a room and a colleague of mine said to me, Jennifer, I'd like you to explain the CRISPR technology to a friend. And he brought me into a room, and person was sitting with their back to me, and as they turned around, I realized with sort of a horror that it was Hitler, and it was actually Hitler with a sort of a pig nose, and it almost looked like a chimeric pig human sort of creature. And it was, it sounds funny in a way to relate that image, but in the dream, it was a terrifying thing. And I really felt real, just, stone cold fear in the dream, and sort of woke up from that dream with a start and realized, kind of had this initial feeling of what have I done, and and that was really one of the things that motivated me to get out of the lab and start talking to people more broadly about the technology, about its capabilities, about the great things about it, but also about things that really required really deep thought and careful consideration and regulation.
Saints, did you guys hear what she just said? Saints. Now, I need to make
Our ability to manipulate genes can be very powerful. It has been very powerful.
This is going to revolutionize human life.
All right. Sorry about that. I had to plug my mouse back in. I'm doing dual work. I'm doing the Lord's work, and I'm doing my job as well. And I'm also trying to eat so I don't fall asleep or get hangry. That's the worst possible thing that can happen right now. Right, wife? Love you. So excuse my smacking. Few things to know here. All right. Everything that she just said in there, I just wanted to make sure that you saw the video of it, and it's not fake. All right. That was Jennifer Doudna, and she's talking about Hitler having a pig face, and kind of like capping it in like a contradictory manner of saying, all right, I don't want this to go into the wrong hands and such like that. And that's why, after that dream, I wanted to make sure that, it was done the right way. But you just saw her before the dream talk about pig organs and being able to use pig parts and things like that to help people. All right. So I want to show you something here. You should already know where this is going. Luke 8:32 says, and there was there a herd of many swine feeding on the mountain, and they besought him that he would suffer them to enter into them, and he suffered them. So we'll get another. That was in Luke. We'll go to Matthew. And the beautiful thing about scripture I love is that there are a few different places you can find, precept upon precept, as it talks about in Isaiah, I believe. This is Mark chapter 5, verse 12 and 13 says, and all the devils besought him, saying, send us into the swine, that we may enter into them. And forthwith Jesus gave them leave, and the unclean spirits went out and entered into the swine, and the herd ran violently down a steep place into the sea. They were about 2,000 and were choked in the sea. All right. I want to show you guys something. This is just one example. Very recent, though. Very recent. The gene-edited pig heart given to a dying patient was infected with the pig virus. The first transplant of a genetically modified pig heart into a human may have ended prematurely because of a well-known and avoidable risk, circa 2022. May 4th. This was just a few weeks ago. Now, mind you, the actual procedure, this took place, I think, what was it, last year or maybe the year before. There was a lot of hoopla about it in the news. But now this guy's dead, unfortunately. All right? I said that pretty cold. This guy's dead, Noah. This guy died, all right? Now, Says the pig heart transplanted into an American patient earlier this year in a landmark operation. Okay, so it was earlier this year. Carried a porcine virus or porcine virus that may have derailed the experiment and contributed to his death 2 months later, say transplant specialists. Um, that's all I needed to know. You know, that's all I need to know. Now, in regards to organ transplants between humans, there are rejections, and people can develop infections. They can have what is it? What is it? The Lord, bring it back to me because I used to know this. Um, hmm, I can't remember, but there's a condition that happens in the body, a very bad condition that happens when there's a rejection of a new organ that's been, transplanted. That type of stuff happens between humans. I don't want to be injected with. I don't want to transplant from anything else outside of that because the odds are not in your favor. All right. We're getting the context here. So CRISPR is a gene editing technology which can alter the characteristics of genes and genetic expression. Genes are responsible for who we are as humans, characteristics that define individuality in all of us. I want to show you something here. Pull this off screen. Work with me now. All right. I want to show you something. Let me show you something from if you ever watched In Living Color. Let me show you something. Fire Marshal Bill. Anyway, um, I'm starting to feel a lot better. Thank you, Lord. I had to warm up some food. I want to show you this. Now, this is the patent of, you know what? Keep coming back to this. Again, like I said before, um, in this patent, basically based off of a lot of research that they did. Um, and these are sites, articles that they reference to use. So CRISPR is in here 6 times. All right. So we see that this is a part of this. Okay. They don't reference these types of things unless the they utilize the research data that's in the papers, the white papers. So I apologize. I'm just eating. I've got to eat. We will go. We were there. Get back on track, Lord. Thank you. Now, do you remember this? From the first installment, all the way back in the in vivo abomination that I did last year, um, the Human Genome Project. The Human Genome Project was one of the great feats of exploration in history, rather than an outward exploration of the planet or the cosmos. The HGP, which is Human Genome Project, was an inward voyage of discovery led by an international team of researchers looking to sequence and map all of the genes together, known as the genome, of members of our species, Homo sapiens, beginning October 1st, 1990, and completed in April 2003. The HGP gave us the ability for the first time to read nature's complete genetic blueprint for building a human being. 1 second. Need to check on this. Okay. Sorry about that. All right. So let's see. Now we got all the way here, if you remember, from all the way back from in the beginning of this series. right when we were going into the Nimrod protein. We got here from Aleph to Tav, Alpha and Omega, the beginning and the end. We've covered the Alpha technology that created the Nimrod Eater gene. I said Eater gene. We've got a few more references to Alpha Aleph to consider. And I want to show you something here. And that requires me to bring up a new window. We are done with that. So bear with me here. One moment while I do a few things off screen. Leave, yeah, because I brought that up. I know. All right. So, a second here. Let me bring up my new one. Take a bite. Thank you guys for being so patient with me. Oh, man. Thank you, Lord. Thank you for patience. Thank you for this wonderful food my wife cooked. Oh, my goodness, baby. I love you. Thank you. Mm. So I've got to wait for these to load up here real quick. Bear with me. 1 second. Opening. Opening, opening, opening. We are 56 minutes in, so I will try to cap this at hopefully in an hour and a half. Lord says the same. Lord says the same. I like to keep these relatively the same amount of time it takes for the average movie to be watched. At least that's what the Lord put in me. So loading up here. I'm gonna look again and just make sure. All right, all right. It's popping up. Amen. All right. So with that being said, everything that we just covered there, we need to shift gears. All right. And I need to show you something. You've all seen this. Probably didn't think this was going to pop up in the research. Like it wouldn't have anything to do with this. We've all seen this. We know what this is, right? Let's take another look. All right. Now, I want to show you something. Let me show you this sometime. Let's see. Where is it at? Where's that? 1 second. This is going to be very relative to what we need to see here. Or please help me find it here. How about this? No, it's not that. Oh Lord, what did I do with it? What did I do with it? It has been a minute. Let me 1 second. Bear with me, Saints. There it is. Take a look at this. All right. Now, what did we just see? We saw this. Black Lives Matter. All right. Black Matter lives. It's in a different order. Now, I had to make this picture. I'm going to tell you exactly why I had to do this and what the Lord revealed to me in this. All right. And how this relates. Sorry, taking a drink. All right. I'm going to show you this. Let me get that back up here. Now, you notice any difference between these? All right. You should be noticing the difference. Black Matter lives. Black Lives Matter. What do you see? Now, throughout this series, we've seen the alphanumeric principles expressed in arithmetic, language, dialect, formulative expression, and equations. Now, in applying proper reading technique and the prioritizing of words while also following the fundamental way we're to see and read problems in order of operation, how does it look to you? How does this look to you? Does this look a little off? Like we're reading it wrong? All right. The thing is here, it looks improper. Improper in the sequence that we've become accustomed to reading it as. Unbolded word, unbolded word. But it begs us to read it in its proper sequence. Remember the coded language, the doublespeak in regards to the enemy. What if I told you that this logo, this saying, this chant that was heard around the world and spoken of by billions of people was again the enemy speaking in code and used as an integral part of invocation and spellcasting? Now, in reading this correctly, again, this is what we're supposed to come up with. In reading this correctly, we begin to see what it truly says and what it truly conveys in coded language. Black matter lives. All right? I want to show you something here. What is black matter? Aha. We see where we're going with this, Saints. Breaking new ground in the search for dark matter. August 7th, circa 2020. All right? Um, the Large Hadron Collider is renowned for its hunt for and discovery of the Higgs boson. But in the 10 years since the machine collided protons at an energy higher than previously achieved at a particle accelerator, researchers have been using it to try to hunt down an equally exciting particle, the hypothetical particle that may make up an invisible form of matter called dark matter, which is 5 times more prevalent than ordinary matter and without which there would be no universe as we know it. So that's the paradigm that they're talking about. The yin and the yang. You can't have the good without the bad. Such and such. Star Wars. Dark side, light side of the Force. Things like that. Um, 1 second. Let me show you this. CERN. Let's see. Is a European research organization that operates the largest particle physics laboratory in the world. And let's see. CERN's main function is to provide the particle accelerators and other infrastructure needed for high energy physics research. All right. I'm going to show you this. Before I show you that, I need to say, um, CERN smashes particles together at atomic forces and at incredulous speeds. The same particles that are the building blocks of our cellular makeup, the protons, the protons, the neutrons, and the electrons. These are what make us up from down at a cellular level, and they're taking these particles and smashing them together at incredulous speeds. Um, this takes place at the most expensive man-made scientific experiment in the history of the world, and they're looking for a particle called the God particle, which they term is the glue that holds everything together, and to open a doorway to another dimension. Got it. Especially when you consider that there's an array of sixes in the logo, as we see here, as we see more fully here. Now, I want to show you this. The Aleph experiment, which we know Aleph to Tav, Alpha and Omega, beginning and the end. Aleph is Aleph is where we get Alpha from. All right. Aleph was a particle detector at the Large Electron Position Collider. It was designed to explore the physics predicted in the Standard Model and to search for physics beyond it. It operated from 1989 to 1995 in the energy range of the Z particle, around 91 GeV. Um, I don't know what the G stands for, but I know the E stands for electron volts. And later, 1995 to 2000. Now, this was the first accelerator run for this facility at CERN and named the Aleph experiment. Remember, Aleph is alpha in Hebrew. Second here, take a sip. Need to show you this. CMS. CMS detector uses a huge solenoid magnet to bend the paths of particles from collision in the LHC. The compact muon solenoid CMS is a general purpose detector at the Large Hadron, or we'll just say LHC, a broad physics program ranging from studying the Standard Model, including Higgs boson, to searching for extra dimensions and particles that could make up dark matter. Again, all right? Although it has the same scientific goals as the Atlas experiment, it uses different technical solutions and a different magnet system design. I need to show you this. Brookhaven National Laboratory, Physics Department. Um, In the early 1970s, the BNL Physics Department reorganized the experimental high-energy physics groups. A vote was held for the name of one of the groups, and the majority chose Omega in recognition of their discovery of the baryon made of three valent strange quarks. I don't know what those are. First seen in the BNL 80 bubble chamber. Um, um, so you see that. Now I need to show you this. The Omega Group. In the early 1970s, the Brookhaven National Laboratory Physics Department reorganized the experimental high-energy physics groups. A vote was held for the name of one of the groups, and the majority chose Omega in recognition of their discovery of the W negative, the baryon made of the three, blah, 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 blah. Now, I need to show you this. Current experiments. Omega Group members are collaborators on the following experiments: Atlas experiment at CERN Geneva, Switzerland, and U.S. Atlas collaboration. All right. Um, this brings us to Atlas. Atlas. From a cavern 100 meters below a small Swiss village, the 7,000-ton Atlas detector is probing for fundamental particles. Is one of the two general-purpose detectors at the LHC, and it investigates a wide range of physics from the search of the Higgs boson to extra dimensions and particles that could make up dark matter. Again, Um, Although it has the same scientific goals as the CMS experiment, it uses different technical solutions and a different magnet system design. I need to show you this. Now, this is the Atlas experiment. I need to show you this. Atlas mythology um, is a titan condemned to hold up the heavens or sky for eternity after the Atlas also plays a role in the myths of two of the greatest heroes, Heracles, or Hercules in Roman mythology, and Perseus. Now, again, the etymology work shows us Hercules goes back to Nimrod. Um, According to the ancient Greek poet Hesiod, Atlas stood at the ends of the earth in the extreme west. Later, he became commonly identified with the Atlas Mountains in North Africa and was said to be the first king of blah blah. I need to show you this. All right. Um, what we're getting here, what we're getting here is an introduction. But again, the enemy, as we spoke of, has many identities. All right, many identities. And if you know about the lowercase g God name game, um, throughout the cultures and um different millennia, you'll have the same figures. Um, you'll have the same figure represented by a lot of different people. Same, but you'll have a lot of the same characteristics and whatnot. And I call it the lower G God name game. Um, Now, what they're saying here, this is something to really keep in mind. Atlas, according to the ancient Greek poet, Atlas stood at the ends of the earth, and was also known as a Titan condemned to hold up the heavens or sky for eternity after the Titanomachy. Now, I need to touch on this. Lord wants me to touch on this. In Greek mythology, this was a 10-year series of battles fought in Thessaly, consisting of most of the Titans, an older generation of lower G gods based on Mount Othrius, whatever, fighting against the Olympians. So two factions of Titans fighting against each other. All right. Keep this in mind in regards to this Titan Atlas condemned to hold up the heavens or sky for eternity after this. This is an artistic depiction of him holding up the world. All right. We are going to come back to this guy in a major way. All right. But the Lord wants me to touch on something. 1 John 2:2 it says, and he is the propitiation, yeah, propitiation for our sins, and not for ours only, but also for the sins of the whole world. All right. Um, I wanna. Oh, I can't touch on that yet. I can't touch on that yet. I believe this Atlas is somebody who well, I know this Atlas is somebody we're going to be covering. All right, but remember to hold up the heavens or the sky for eternity after this. All right, especially in relation to this verse. We're going to go forward now. I want to show you this. Okay, the Lord is telling me to touch on it. Okay, Atlas, who we just talked about, and we're going to talk about this other character here. I'm going to bring up. Atlas is in relation to Abaddon or Apollyon, and a mockery of the Most High in regards to this being a depiction of Atlas, in regards to 1 John 2:2. All right, and I'm going to show you later on, but we have to keep in this relation of Atlas being, and I'll go ahead and pull it up, Apollo. All right, Apollo or Abaddon, who the Bible says Abaddon in Greek is Apollyon. All right, keep that relation in mind. Okay. What we need to go to next is what's called the Hippocratic Oath. Now, the Hippocratic Oath is an oath of ethics historically taken by physicians. Okay. I want to show you something. In the original, well, it says the earliest surviving copy of the Hippocratic Oath. I understand it's gone through different variations and over the years. The earliest copy says, "I swear by Apollo, healer, by Asclepius, by Hygeia, and by Panacea, and by all the gods and goddesses, making them my witnesses that I will carry out according to my ability and judgment this oath and this indenture." All right. So the physicians have always look when you swear an oath, no matter how many different variations there are of it, you're still swearing by the same thing. It was first and foremost said, "I swear by Apollo," and we know Apollo to be Apollyon, aka Abaddon. All right. In regards to this verse here, Revelation chapter 9, verse 1. And let's see. And the fifth angel sounded, and I saw a star fall from heaven unto the earth, and to him was given the key of the bottomless pit. Revelation 9:11 says, "And they had a king over them, which is the angel of the bottomless pit, whose name in the Hebrew tongue is Abaddon, but in the Greek tongue his name hath his name Apollyon." Um, Now Apollo, excuse me. Now getting back to that Atlas, the sins of the world being attributed to him. We will revisit that again. We're going to revisit it later in the series, but please keep it in mind. All right, we've got to go forward in this Atlas here real quick. So the etymology of Atlas. All right. Let's see. I need to reference in words from Greek such as abysmal, adamant, amethyst. Also partly nativized as a prefix of negation of asexual, amoral, or agnostic, the ancient alpha privatum, denoting want or absence. All right. Um, Again, in old, we get it to alpha, alpha privatum. Um, Now take notice this right here, this asexual reference. Keep that in mind. We will revisit that. All right. Um, we need to go back to we need to go to this. Now, Atlas in regards to anatomy. All right. Um, Second here. Where's that? 1 second. All right. Now, the atlas and axis are important neurologically because the brainstem extends down to the axis. All right. Um, Now, the brainstem pathway leads to the blood-brain barrier and access to the brain, which we've discussed a few times over. We will have to revisit this again in just a moment in regards to atlas and mapping, but we will get back to that in just a moment. All right. Um, and that connection to mapping, if you haven't caught it already, as far as Atlas, which you know what I'll go back to here. All right. Let's see. Now, let's see. It's in relation to maps here. So the term atlas has been used to describe a collection of maps since the 16th century. All right, as a point of navigation and whatnot. So you see it used as terrestrial maps, nautical maps, a few different things. All right, in regards to mapping and sailing the seas and whatnot. Um, I need to go back to, let's see. Got it. Okay. So we need to go to, we need to go here. We've covered the alpha. We've had a reference of omega. We need to go ahead and take a look at omega. This is Omega Therapeutics. Now notice the emblem. Notice the emblem. That emblem should be setting off some red flags in your discernment there. Um, Omega Epigenomic Programming for Precision Genomic Control. So again, we're getting another ode to editing software. All right, a gene editing software. Now, we go here, October 15th, circa 2021, or that's when it was updated. The man behind Moderna on what's next in biotech. All right. Um, this, let's see, 1 second. So the funding. So the funding for, where is it at? The guy who was the big wig wallet behind Moderna. Um, here it is. What's next? What's next here? What is next here? 1 second, Saints. Where is it at? Startup. Let's see, where was it at? Oh, that's what it is. Okay. So basically in this article, dang it, it happened again. Okay. Basically in this article, this guy's being interviewed. He's was the big wig wallet behind funding this company. And his next startup company that he was helping to fund is this Omega Therapeutics. Um, and we're gonna go here. So notice these guys are moving and grooving. All right. Omega grabs 126 million to bring genome tuning cancer treatment into the clinic. This is March 30th, circa 2021. Here this is just another article for the same thing. Here they're snagging 85 million. This is July 29th of circa 2020. Um, Now these folks, all right, especially in their relation to uh, Moderna, okay, these folks are the next ones, especially in regards to this cancer research and genome editing. You need to keep your eye out for these folks. They're groundbreaking genomic research and cancer treatment, and I believe in my spirit that this will have a big part to play in things to come. Now, earlier on, we discussed graphene and the electromagnetic spin properties it relies on that produces a magnetic field. With the CRISPR and Alpha and Omega gene editing companies specializing in gene editing and expression. This is where we have to bring in something called magnetogenetics, and that will be covered in our next video. I thank you so much for watching this one. This one was a tough one to get through. It's late. I'm at work. I needed to eat in the middle of it. I appreciate your patience, Lord. Thank you for making this edifying. Thank you for uh, counting this worthy, hopefully, Lord. And thank you for considering me and being a worthy servant and doing your will. Again, God bless you all. Love you all. We will go ahead and wrap this one up, and we will be back for part 4. All right. God bless you all. See you soon.